And now for someone completely different. Some of what I'm going to talk about, overlaps a little bit what David discussed, but that's okay. But I'm going to try, my title is Continuing Evolution of Genetic Toxicity Testing at the EMGS. I'm going to be talking more about the development of the society and the science, because they went together. They're not separate. And this is going to be my opinion. My disclaimer is this is my opinion. It is my memory, my recollections, and it's really brought back for 50 years because I joined the society in 1969. So it goes back then. By the way, what you're seeing there next to me was the latest in the electronic colony counter. It's called the Quebec counter, and it preceded the colony counters that we're using now by about 5 or 6 years. But it worked. Okay, David mentioned the Crow report and the Marac DHEW report. They're very interesting. What Jim Crow and his society and the committee recommended was tests for mutagenicity be a routine part of testing of chemicals. This was a new concept at that time. And what the Marac Commission report was specifically for pesticides, and both of these were oriented towards germ cell mutagens, towards heritable effects. The Marac Commission report was interesting because one of the recommendations was test all chemicals in 3 mammalian systems. The dominant lethal test, which most people will recognize, host-mediated assay, which very few of you will recognize, and in vivo cytogenetics, which we're doing now. The host-mediated assay was very interesting. At the time it was recommended, it had not yet been published. When it became was published also in 1969, there were 4 chemicals that had been tested. The assay was you injected Ames is Salmonella strains into the peritoneal cavity of a mouse. Then you administered the test chemical by a different route, intramuscularly or orally. Then 3 hours later, you removed the bacteria from the peritoneal cavity, diluted and plated them for mutation frequency. When the publications came out, there were 4 chemicals published. All 4 were positive. 2 of them did not need metabolic activation, which is the whole point of the host-mediated assay. One of them that needed it was dimethylnitrosamine. Another one was cyclosin, which needed metabolism by the intestinal bacteria, so it only worked orally. But this was recommended without any data. And even though there had been chemicals tested that were negative, they were not published. But this was the state of the art at that time. Okay, moving on, one thing that David didn't mention, but I think is very important. In 1964, Frits Sobels in the Netherlands started Mutation Research Journal. Why it was important is because this provided, when EMS started and when mutation research studies started, it provided a venue for publications in mutation research. Someplace you could go to if you wanted information about mutation research. And you see that in 1969, Medical Mutagen Society started. 1970 at FDA, Marvin Legata was the head of the genetic toxicity branch. This was a recognition by FDA at that time that genetic toxicity was an endpoint worth studying, worth mentioning. And using that, Marvin awarded the first contracts for testing chemicals for mutagens, food additives for mutagens, in 1971. We did not have any standardized protocols, we didn't have any standardized other stuff, but he awarded in 1970. And to show you where the science was, it was 1970 that the reverse transcriptase was first defined. And in 1972 is when Sanger proposed his DNA sequencing. So this was the state of the science at the time. And another important thing in 1972, the US Congress passed the Cancer Act, which provided a lot of money, not only for clinical cancer, but also for research, which resonated down through the rest of the field. And in 1973, this is when Bruce Ames essentially shocked the world and said, here is a quick and easy test to test chemicals for mutagenicity, allowing for metabolism, and also letting you know whether you were looking for a frame shift mutagen or a base pair substitution mutagen. And this started the shift from emphasis on germ cell mutagens to an emphasis on carcinogens. And at the same time, at the same year, about the same time that Bruce came out with his publication on the method, Bryn Bridges in England published a tier system for mutagenicity testing. Again, without specific chemicals. It's different from what we're doing now in regulatory toxicology, but it may not have been that good. It's better, I think. In his testing, if you had something that was positive in vitro, you went immediately to risk assessment. Don't bother looking at in vivo mutagenesis. And since I'm running out of time, I have to speed up here. So a number of important things. The most important thing is in 1976, the US Congress, in a spurt of activity, they were more active and more proactive then, in that 1 year, than they have been in the last 15 years. They passed the Toxic Substances Control Act, which required EPA to start testing using mutagenicity as an endpoint. And they said that NIEHS must start a mutagenicity testing program for carcinogenesis. Why? Because Bruce Ames in 1975 published that hair dyes are mutagenic. And Congress somehow learned about that paper, and in their deliberations said, because we are now concerned about mutagens and carcinogens in the environment, we want NIEHS to start testing. And this test program later became part of the National Toxicology GTOX testing program. And there was another DHEW report in 1977 that was started in 74 that also addressed mutagenicity testing for heritable translocation, for heritable mutations. EPA came out with its first testing scheme in 1978, and OECD started in 1979. It was a very exciting time to be in GTOX. That's David Brusick talking, and I was really concentrating. Okay, this was interesting. This was the first proposed EPA TSCA testing scheme. It was a menu. Choose 4 from column A, 4 from column B, 2 from column C. And you notice one of the tests that you could choose from was the mouse-specific locus test. No more, at any time, there were no more than 3 laboratories in the world doing that test. And mega animals, mega millions. EPA never asked for that test because it wouldn't have been done. They refined the scheme a few years later, but still was asking for the mouse and for the specific locus test. But they had separate channels for carcinogenicity and germ cell mutagenicity. And finally, it got better and better, and this is more appropriate by 1991 to what we're doing now. One important discovery during the 1970s is, we used to think that mutagens were very rare substances. In the 1970s, we realized they're everywhere. Synthesized by chemists, by Mother Nature, in the environment, produced when we're cooking or processing foods, and we produce them in our own bodies. And I want to go into a little now evolution, the EMGS in its journal. And, when it started, as David pointed out, it was more identifying mutagens and some basic mechanism studies. We didn't have the equipment, we didn't have the knowledge. Now it's morphed into mostly mechanistic studies, and studies going beyond just, is the chemical a mutagen? And the journal is the same. If you look at the early programs of the EMS, half the talks are, is the chemical a mutagen, or I have a new mutagen, or I have a new test system to show that something is a mutagen. And incidentally, just as an aside, David pointed out that the one female speaker at the first meeting was Jackie Barrett. Nobody knows her name now, but she was probably one of the most important people in keeping thalidomide off the US market. You know, the Europeans had approved thalidomide as treating women in pregnancy. Jackie Barrett was a teratologist who was using chick embryos, and she found chick embryo teratology from thalidomide, and I believe it was a wing formation, which paralleled the later Phocomelia in humans. And her data was used by Francis Kelsey, who was a reviewer at FDA, and between the two of them, they kept it off the market. And then a couple of years later, it was discovered that thalidomide was causing all these problems. So she was a good choice for speaker. Okay, this is just a quick review of where the EMS was in the 60s and 70s, and where it was in the 90s and currently, moving from descriptive to understanding what's going on. And this is the membership over the years that I've been able to put together. It's a little spotty, partly because we've had about 4 or 5 different management companies, and the records were kept first on paper, then on, more the early computers, the later computers. So we're still trying to accumulate all the data on it. But at its height in the 80s, there were over 1,000 members. Now it's down to a few hundred. And I think there's some good reasons for that. The field has matured. We don't have as many labs looking for new test systems to identify the next new mutagen. In the 80s, there were departments of genetic toxicology in universities and in companies. And because the field has matured and condensed, gene tox is now a part of toxicology itself. It's not as much of a separate field. And also, I think it's important is, a lot of people join society so they can get the journal. If you work for an organization, a university, or a large company, you don't have to join the society to get the journal. You just get online and you have your free copies. So it's another reason for people not to be members of the society. And where is genetic toxicology and genetic toxicity going? The next 8 or 10 speakers will tell you exactly where it is. And I could say I've been a member since year 1. I'm not a charter member, but I had the good fortune when the first week of working in gene tox, I was hired by Marvin Legator, but I got to meet Fred DeSayer, Heinrich Malling, Gary Flam the first week, and within a few months I was also meeting Alex Hollander and the others. So I've had a great life in the society, and I encourage anybody here who's not a member, please consider membership, because it's a very collegial group and a very understanding group, and a group that is willing. From the very beginning, it wasn't Dr. Hollander, it was Alex. It was all a first name, very casual basis, and very interactive, and there was no distinction between people who had a bachelor's degree, master's, or PhD, or an MD. So that's it. Thank you. You'll hear a lot more later on about the history of the society, but this is my take on it. Thank you.